Today, we'll be discussing epistasis. Our genetic information that makes us who we are is encoded in chromosomes located all throughout our bodies. On chromosomes, there are genes. These genes are what give us the unique traits such as eye color and finger length. All of the possible expressions of a gene, such as blue versus green eye color, are known as alleles. However, sometimes genes that control, for example, eye color and finger length may interact with each other and either mask a certain expression or change an expression drastically. A genotype is the genetic information in our chromosomes, and our phenotype is the physical expression of them. Imagine that you're an employee of Chick-fil-A. Alright, so you could either go in to work and have a really good day, a bad day, or a horrible day. But it really depends on which managers are there and if the owner is present. So, let's look at the managers. There's Mike and there's Stephanie. Mike is really helpful. He's very adaptable to changes. He doesn't micromanage the employees. But on the other hand, Stephanie does, and she always makes the workplace kind of tense and kind of scary at times. However, something worse could happen. Imagine the owner, Miley, came in. She is so mean. Epistasis is just the interaction of one gene on another. Put simply, in this case, it's the interaction of the owner gene on the manager gene. No matter what manager is present, if the owner is present, so loci 1, then it's always a very bad day at Chick-fil-A. Everyone starts with having a good day, and in order to get to the bad day intermediate, there must be a lowercase aa, which is manager Stephanie, or a capital B, which is the owner within the genotype. However, if there's an uppercase A, then this could potentially stop the transition from good to bad day. The large T-like figure in purple represents an inhibition of the pathway at that specific location. In order to go from a bad day to a horrible day, there must be a big B present in the genotype. If there are two little b's, then this pathway is inhibited and would stop at the previous pathway transition. This is an example of single dominant epistasis. When the dominant or capital B is present, it masks all other possible expressions and leads to having a bad day, and then leads to having a horrible day, no matter which allele is present in the A gene. First, we would need to set up the possible genetic combinations that each of the parents could pass on to their offspring. The two parental genotypes are shown at the top, separated by an X. Each parent in this case could pass on 4 possible combinations of genes to their offspring. Each parent can pass on one allele at a time for each respective gene. The possible genotypes at the top are for the parent listed first, and then the vertical column represents the possible combinations that the second parent could pass on to their offspring. We will now need to write out all the possible genotypes for offspring. We will look to see where a specific column and row intersect and write those possibilities out. Now, let's categorize all of the possibilities into 4 groups: big A and big B, big A with 2 little b's, 2 little a's with a big B, and all lowercase a's and b's. We can now do the same thing with a different cross, as seen here. Now we can look to our fictitious pathway to see which phenotypes are associated with each of these genotypes. Here are the final ratios of phenotypes for the 2 crosses, horrible day to good day to bad day. But no matter the day, it's always my pleasure.